Hello and welcome to an introduction to my project. On here is a full presentation of the master's thesis I'm working on. There were of course a few details I forgot to mention during the initial presentation, and those will be clarified in the end in an addendum. Now let's get started.
There we are. All right. All right, let's get started. My name is Brandon Singh. I'm currently working on this project, a QTL analysis of cadmium accumulation in flax. My supervisor is Dr. Turan, and Dr. Megan House is my main advisor and constant source of advice. Thank you to Megan. Wave to her back there. All right, the experiment will test the hypothesis that low cadmium accumulations controlled through polygenes or quantitative inheritance. And QTL analysis will identify the genomic regions associated with low cadmium accumulation within flax. The goal is to identify QTLs associated with cadmium concentration in seeds of the flax rills and to validate SNPs associated with cadmium concentration in flaxseed. More on that later. There's also some nice to haves. These are little tiny mechanisms I'm hoping to test out if data is available. If it's not, it's not, but this is like the flat screen TV equivalent of the experiment. Nice to have, but not necessary. One is Dr. Kenishuk's model of high protein of cadmium uptake. This means that Dr. Kenishuk in the past associated high cadmium with high protein, and if there's enough protein data or any protein data, that we will test that out. And the other one is Dr. House's personal model of early maturity reduces cadmium uptake. And if there's enough data, I'll be testing that one out. Now, there's two different flax types in Saskatchewan being used. There's fiber flax and seed flax. Fiber flax is used for fiber, and seed flax is used for its seeds, particularly its oil and edibility. The meal can be utilized in making different types of baked goods. It can be eaten fresh in cereal. And the oil is quite good for both industrial purposes and just for cooking and salad dressings and whatnot. Now, why flax? High protein content, good oil content, has phytoestrogen lignans, which actually can help combat cancer. Not really combat it, but prevent it a little bit, reduce the likelihood of it happening. Has a high levels of different vitamins and a high level of alpha-linolenic acid and other omega-3 fatty acids. There's a bunch of drawbacks. The key one we're talking about is cadmium. Why is this important? How do we get here? Well, cadmium, like most other heavy metals, causes various health issues, as you can see by this unique Creative Commons picture here. Neurological problems, lung disease, cancer, a whole bunch of other problems as well. And there's also new EU regulations. There's a new EU requirement for cadmium, 0.5 milligrams per kilogram of seed. Now, how did we get here? Well, one of the biggest problems is that in the 20s until around the 50s, there's been a heavy amount of industry, smelting and whatnot, and because we didn't have the filters back then, all the cadmium that was burned off just came down in acid rain. And of course, we were also one of the major cadmium manufacturing processors back in the day. And as time went on, people just sort of took the wastewater and splashed it into the rivers, lakes, and everywhere else. So Saskatchewan prairie soil and all the prairie soils in general, and as well as Quebec, have varying degrees of cadmium. And the killing germplasm accumulates cadmium above the certain specified EU limit. Now here's the plant materials. We crossbred this guy, a derivative of MP30, an Indian landrace made through pure line selection. It has low cadmium concentration in its seeds and it's an experimental line, and the heir apparent is bison. A major heirloom cultivar utilized for its fusarium resistance and iodine, but has high levels of cadmium concentration. Through this process of crossing, we made 120 different lines, rills, through crossbreeding bison and FMC74 in 2023 to 2025. They're brought to the F6 generation under speed breeding conditions under single seed selection. The rills were grown under both Phytotron conditions and field conditions, with 3 reps on each. Kernan soil was used in each trial. And the phenotyping that we'll be using would be ICP, MS, and NIR. And the key traits being measured outside of the level of cadmium in the seeds would be days to maturity, days to flowering, yield, 1,000 seed weight, and all the phenotypic data will be analyzed in the R program. R will be used to analyze the data. You will first just sort of visualize the data, make sure everything's set to go with ANOVA, compare the parental means and all that jazz. And then the linear regression will be done to compare the variation between indoor versus outdoor, determine the possible relationship with cadmium and protein, or days to maturity. This is nice to have, not required, but basically we'll just be comparing the parental means through linear regression. No, sorry, compare the parental means of cadmium using linear regression. And QTL analysis and GPS, the DNA was extracted from each rill and parental line, as well as CDC buffoon, using the commercial DNeasy Plant Pro DNA extraction kit under standard protocol. And then this nice little company, LGC, will be making a genetic map, linkage, the full 9 yards, using SNP map markers generated through GBS. And you'll analyze the phenotypic data we'll gather and the genetic data we gather using R, specifically the package QTL2 and ICI mapping. The goal is to validate these markers right here. These are markers previously discovered by Dr. Sylvia Cloutier and Dr. Frank Yu. These were using the data obtained via AAFC Ottawa. This was derived from a diversity panel. There are 14 markers that are associated with CD content, and we're going to validate which ones are in the population we've created and which ones are not. Now there's no physical data, unfortunately, and this is thanks to indeterminacy, one of the banes of every flax grower's state of affairs. As you can see on this picture I got from stock footage, there's a great deal of flowers and seeds because our population generated extensive numbers of flowers due to the fact that we had really good weather conditions near the end. And the population was surprisingly highly indeterminate. What does this mean, you may ask? It means that harvest was delayed a little bit. I expected the harvest to be around late August, early September. It was not that. and that meant that we couldn't get as much data as I wanted, and that is why I'm kayfabing a little bit. Outside of this, there's also a couple varieties, a couple lines I found that show promise. Just on a cursory inspection, no scientific basis on that. I'll be analyzing that data sometime in January, but there'll be a few lines I'll be keeping a close eye on. And now that we're finished, I'm hoping for questions.
You're studying most of the phenotypic data and also yield-related data, but you are analyzing the relationship of cadmium with only days to maturity and the protein.
The actual goal is, of course, to validate existing QTLs. The mechanisms de jour is comparing the cadmium content with days to maturity and protein, if the data permits. But the key thing is validate the pre-existing QTLs. This is the tail end of the project. Two mechanisms that I'm tinkering with are nice to have. They're like the flat screen TV. You can do without it, but it'll be nice to have. We're gathering the data. The parents have also been genotyped alongside the many RILs and CDC buffoon. All that genetic data has been sent to the company, LGC, based in Germany, and that data set is being built as we speak. And then we'll go through the entire IC mapping and that data set and merge it together. We don't actually have the SNP chip because it costs too much money, as I learned from Dr. Turan. So we're just sort of comparing the locations. If we are really lucky, we may find some novel locations, but that's again a nice to have, not the main goal. The main goal is to find existing markers in these specific areas. Find basically QTL spikes around here. Just to validate. This is just a validation study in practice, but there's a few other things that are nice to have that I'm hoping to test. But if I can't test them, I can't test them. It is what it is, and that's basically how it goes.
Maybe because that's the only link we have.
Thanks, Brandon. I enjoyed your talk. How long have you been in your master's program now?
This is my second year.
You're enjoying it?
Yep. Having fun. I enjoy talking, and I enjoy writing.
Good thing combination. Couple of questions for you. I was really interested in your hypothesis about it being under multi-genetic control. And have you dug into the literature at all to understand the mechanisms of cadmium uptake in other species like rice? Obviously accumulates cadmium.
Yes, actually.
Durum wheat accumulates cadmium. So I was a bit interested in how you came up with the hypothesis about it being multi-genetic, given that in those species it's generally a single gene.
That seems to be only the case in monocots and dicots, like say Arabidopsis, radish, canola. And I think even sunflower and many other dicot species is all polygenic. A bunch of different hundreds of genes controlled by various QTLs. I think in Arabidopsis, it's about 5 QTLs controlling the variation in cadmium uptake. In rash, I think it's about 8. And it seems to be the case for, it seems to be the same case for sunflower and other species like soybean and whatnot. Because I think there's actually different mechanisms. If memory serves me, if my brain serves me correctly, wheat concentrates the cadmium into a flake leaf, but in the case of Arabidopsis and radish and canola, it's basically sequestration into the root and exclusion from the root using different mechanisms controlled by jasmonic acid.
See, that's the interesting part, is actually Durham, I'm a Durham breeder, so it happened. Kristen, who's here, actually cloned the G. When she was a graduate student, she was in the same place you are, but she was giving the same seminar and asked her the same questions, but we find that in Durham, it's sequestration in the root, much like the mechanism that you're talking about in dicot. So I find it interesting that, you know, in the dicots, it has this sort of complex pathway. In the monocots, relatively simple for the same physiological mechanism. Anyway, thank you for that. Appreciate it.
Thank you.
Great.
Now on to the addendum. Cadmium accumulates in the seeds, which is a primary edible part of flax when consumed by people and by animals. The end goal of this project is to find a lineage within a biparental cross made that can be used to breed a variety of flax that has little to no cadmium in its seeds. Right now, the 2 mechanisms I mentioned are not being tested yet, and most things have not been tested yet because the indeterminacy forced the use of a herbicide to dry things down, and this meant that the remaining of the drying down the seeds was delayed for a little bit longer than expected. By the time this video has been released, the raw data should have been all collected, or at least should be close to being fully collected. Generally be the start point of data analysis that will culminate in the results we seek. What covers everything. Thank you for watching.